everyone, this is Lad from Astrolabe Diagnostics, and today I'd like to discuss a very tricky subject in the context of high-complex cytometry, the question of p-values and statistical significance. And one of the common questions we get here in Astrolabe is, how do we get a p-value of 0.5 or lower? We're running an experiment, we're looking at different cell subsets, we're running differential abundance or differential expression analysis, and researchers often look for this magical number of 0.05. And in fact, there have been numerous publications and discussions about p-values and significance, and I think that one notable publication is this XKCD post that showed up on the internet a while ago, and it includes these different ways to treat p-values where it can be highly significant or significant. My favorite one is highly suggestive significant at the p lower than 0.1 level. I mentioned that this is a publication of sort, and I'll explain why in a sec. But when doing these kinds of analyses in the context of mass cytometry or flow cytometry, there's two problems you should be aware of. The first one is low sample count. High complex cytometry is expensive. Antibodies are expensive. Reagents are expensive. Instrument time is expensive. There's a limit to how many samples you can run. Furthermore, it's possible that you just don't have more samples. If this is a rare disease, if these are patient samples, if this is a mouse study with, let's say, 5 or 10 animals, you have a low sample count. And with a low sample count, there's a limit to how significant, how low your p-value can become. And thankfully, there is a recommendation from the American Statistical Association that discusses just this. The paper was published in 2016, and there is a commentary in the, I think, the Journal of Anesthesio, I can't pronounce it, I'm so sorry. This journal over here. And by the way, I was referring to the XKCD post as a publication because they actually include it in the commentary. And these are two fantastic papers. If you're doing any kind of science, I strongly recommend you read them. I'm going to highlight 2 points here. The first one is recommendation number 3. Scientific conclusions should not be based only on whether a p-value passes a specific threshold. So you should not worry on whether your p-value is 0.05 or lower. More often than not, your p-value cannot be 0.05 or lower. Instead, proper inference requires full reporting and transparency. If your p-value is 0.03, just report it as 0.03. If it's 0.7 or 0.15, just report it at 0.7 and 0.15. By the way, I've seen at least one Cytometry paper who is explicitly using these as a reference to explain why they're still reporting the results even though the p-value is higher than 0.05. It's still interesting, it's still important, it is still biologically meaningful, even if it does not pass a statistical significant threshold. The second thing I want to talk about, and this is a bit of a ghost for many researchers, is multiple casting correction. When you're running these kinds of high complexity studies, you have 20, 25, 30 markers, you're finding a large number of cell subsets. Then you're running a statistical test for each one of these subsets, or maybe even for each marker within each subset. Let's say you're looking at naive CD8 T cells and memory CD8 T cells and naive CD4 T cells and so on, and then each one of them you're looking at PD1 and CTLA4 and interferon gamma and so on and so forth. Each of these is a separate test. And when you're running multiple tests, you need to correct for them. So this is a common use case where you run differential abundance analysis, you're looking at naive CD4 T cells and you see a p-value of 0.03 and like, oh, this is great. However, you've actually analyzed more cells than this, and you need to consider all of the tests. So what I'm showing you here are different cell subsets in the x-axis and different p-values on the y-axis. And we have this one subset that has a p-value of 0.03. However, we've actually tested multiple cell subsets. So we have a wide range of p-values, and we're looking at one of them, which happens to be the lowest. However, when you're doing multiple tests, you need to correct for them. There's different ways to do it. The most common one seems to be false discovery rate. And briefly, very hand-wavy, it inflates p-values based on the number of tests. So if we would take these p-values and apply the FDR for them, the BH correction to be more precise, this is what we're going to end up with. So these are still differential subsets, but now the y-axis, instead of being a p-value, is called a q-value or an FDR score. In many ways, it's similar to a p-value, but this is F-jet correction. And in fact, the multiple testing correction completely washed out the result of our test. This is not surprising because these numbers were actually generated at random. So in this case, the multiple testing correction is entirely right. The fact that this is a p-value of 0.03 is pretty much meaningless because all of these numbers are random, and when we test it, we see it goes away. So in addition to having a low sample count, which already gives you higher p-values, you need to correct, which is going to wash that even further. So what can you do? First of all, don't look for statistical significance. Instead, explicitly report the different statistics that you see. Medians, fold changes, p-values, FDR scores, just include all of them. It's very easy to pick the right numbers for the text and then just put a whole table, a huge spreadsheet in the supplementary material. Remember, if you have more markers, you have more subsets, and that means you have more statistical tests, which means multiple testing correction is going to shrink your p-value even further. You need to remember to do the multiple testing correction, otherwise your reviewer will be sad. And once you do, just remember, the Q values are going to be high, especially if you have, let's say, 10 to 15 samples, you're looking for 20 different subsets or 30 clusters or what have you. Just forget about it. It's not going to be statistically significant in the 0.05 meaning of the word. Instead, focus on biologically relevant measures. Talk about fold changes, show the figures, show the data, try to go to the biaxial plots. If you can show, if you're talking about a given difference, then you can go to the biaxial plot, not the t-SNE map, the biaxial plot, and show that. That would be ideal. So that's it for p-values. The usual sales spiel: if you are facing issues with your statistics, if you need help analyzing your data, please reach out to Astrolab Diagnostics. We're happy to help. And if you have any questions about p-values. Thank you for your time and have a good day.